in our genes directs us from behind the scenes. The words within it shape life's destiny. Hidden in your DNA is your genetic dossier. It tells your future and your history. How traits get passed from parents to a child is something that has kept us so beguiled. Cracking the code, genetic mysteries to unfold. Cracking the code, genetic secrets will be told. Cracking the code, genetic mysteries to unfold. Cracking the code, genetic secrets will be told. Cracking the code.
In the 1850s and 60s, Gregor Mendel's experiments with pea plants led him to the realization that hereditary traits are determined by pairs of factors that we now call genes. But there was another route to the same destination, through a microscope. Both the microscopes and the observations through them were mostly made in what is now Germany. First came the microscopic discovery in the 1830s and 40s that plants and animals are composed of cells, the basic unit of all living things. It was also noticed that within the cell is a central structure, which was termed the nucleus. Then came the realization that a plant or animal grows through the multiplication of its cells. In the 1850s, this led to the cell theory of life, which the German scientist Rudolf Virchow spelled out in his famous statement, "All cells derive from pre-existing cells." Improved technology in lenses and in dyes that selectively stain parts of the cell led to further discoveries about the nucleus. In the 1870s, German scientists began to notice thread-like shapes in the nucleus, which were later termed chromosomes after the chromo or color they take up. They were aided by a 17-year-old whiz kid from England, William Perkin, who invented a new aniline dye. They also discovered that the threads split and move apart just before the cells divide into two. This process was called mitosis, which means thread-like. Mitosis is how nucleated cells multiply, creating new tissue and allowing an organism to grow. Prior to mitosis, the chromosomes are too thin to be seen. Then each chromosome doubles itself and the two copies condense and become visible. They are tied together at a midpoint called the centromere. Outside the nucleus, an apparatus called the mitotic spindle begins to form. It consists of two poles from which filaments spread out and surround the chromosomes. Filaments from both poles attach themselves to each of the centromeres. As the poles move farther apart, the double chromosomes line up with their centromeres in the midline. The centromeres then divide, and the filaments pull the two sister copies toward opposite poles. In the final phase, the cell begins to divide. When the process is complete, there are two daughter cells in place of one parent cell. This striking behavior during mitosis led scientists to suspect that chromosomes might have something to do with heredity. It was also noticed that the various chromosomes come in pairs and that their number differs from species to species. But within each species, it stays the same. They began to see how chromosomes are transmitted from one generation of cells to the next as an organism develops and grows. But what wasn't clear was how the chromosomes of the two parents combined together to create the very first cell. Sexual reproduction was still a mystery. One obvious problem was that a simple combination of cells would double the number of chromosomes with each new generation. So attention now turned to the germ cells, the egg and sperm, and their collections of chromosomes. It was soon noticed that the large eggs and tiny sperm have only one set of chromosomes instead of the usual 2 of each. Then, while studying sea urchins, whose egg and sperm cells conveniently meet in the outside seawater, another German scientist noticed that during fertilization, the nucleus of a single sperm cell fuses with the nucleus of an egg cell. So it was now apparent that sexual reproduction is a 2-step process. In the first step, producing the germ cells, the number of chromosomes is reduced by half. In the second step, fertilization, 2 parental germ cells join together, thereby restoring the original number. This mechanism solved the problem of how a constant number of chromosomes is maintained over the generations. And it strengthened the case that the chromosomes are indeed the carriers of heredity. The chromosome-reducing step that kicks off sexual reproduction is called meiosis, after the Greek word for reduction. Unlike mitosis, meiosis involves 2 divisions, at the end of which 1 full cell is transformed into 4 germ or half cells. It starts off like mitosis, as seen here under polarized light. But something different occurs when these double chromosomes line up prior to cell division. They form 2 rows, not 1. The 2 strands of one double chromosome facing the 2 strands of its matching partner. Now begins a kind of genetic tango. The opposing strands of the 2 chromosomes wrap themselves around each other as their limbs crisscross and touch. This is a critical moment in sexual reproduction. It is during this early phase of meiosis, when the chromosomes pair up, that genetic material is traded between them. It is this process of genetic reshuffling that makes each individual unique, and we will examine it in more detail later on. The 2 sets of matching chromosomes are then pulled apart and 2 new cells are formed. These daughter cells each have 1 set of double chromosomes, unlike the daughter cells in mitosis, which have 2 sets of single copies. The amount of genetic material is the same. What is different is how it is organized. In the second stage of meiosis, each of the daughter cells undergoes another division. The double copy chromosomes divide into 2. The sister copies are pulled apart, and 4 new cells are formed, each containing half the original number of chromosomes. These are the germ cells. Meiosis happens only within the specialized organs that form germ cells, the gonads. In animals, those are the male testicles and female ovaries. In flowering plants, it happens in the anther of the male stamen and the ovule of the female pistil. Mitosis, on the other hand, takes place in all other tissues.
If it's the secrets of life that you seek, then through a microscope you must peek.
Mendel did wonders just using his eye, but to really see, you must magnify. You can't help but notice we're nothing but cells, but where in the cell does heredity dwell? The nucleus, that's where it hides. You don't see much until it divides. When chromosomes enter new phases, split into 2. And that is the basis of sexual transmission, which always engrosses our feverish minds. But it's only meiosis, reducing the chromosomes 50%. So when egg and sperm meet at that blessed event, the chromosomes form 1 full set. Just 2 of each kind, that's the best bet. And that new cell begins to grow, multiplies into an embryo. The explanation of this growth is it's due to a process we know as mitosis, and that's what our microscopes helped us determine. In Germany, where all the germ cells are German.
The German scientists who discovered meiosis and mitosis actually knew nothing about Gregor Mendel. His now famous paper came out in 1866, but almost nobody read it, and the few that did couldn't appreciate it. Mendel figured out that the units of heredity come in pairs, and that before fertilization, these units segregate out, so that an egg or sperm cell contains only one of each. But Mendel had absolutely no idea of where to find these hereditary units. He was as ignorant of chromosomes as the microscopists were of his laws of heredity. The stage was now set for a momentous collision of ideas. Mendel's discoveries about heredity and the new facts about chromosomes were about to meet. When it finally happened, this collision gave birth to a brand new science: genetics. In 1900, 16 years after his death, Mendel's neglected paper was finally discovered in a most peculiar way. 3 different European scientists each claimed they independently did plant breeding experiments similar to Mendel's and discovered the same laws of heredity. Then, in another amazing coincidence, each of them also claimed that afterward they stumbled upon Mendel's paper and realized he had beaten them by 35 years. It seems more likely that the second coincidence happened first. Recognizing the importance of Mendel's long-lost work, each probably hoped to reproduce it and then take the credit for himself, until they realized the others were onto the same trick. Having caught themselves in the act, they had little choice but to give Mendel the credit he deserved. Mendel's theory that pairs of factors segregate apart into different germ cells corresponded nicely with what was seen during meiosis, where pairs of double chromosomes separate themselves during the first stage of germ cell formation. Since chromosomes act like Mendel's factors, it seemed only logical that they must carry them. This union of Mendel's laws of inheritance with the new microscopic findings about chromosomes was called the chromosome theory of inheritance. It quickly gained acceptance among early geneticists who began to refer to Mendel's factors as genes. But which chromosomes held which genes? And since there are clearly more genes than chromosomes, how are multiple genes arranged within the same chromosome? The next step was to look for differences among the various chromosomes. In 1906, 2 unusual chromosomes were noticed in insects. The larger of the 2 was termed X, and the smaller one Y. Nettie Stevens and Edmund Wilson noticed that females always have 2 copies of the X chromosome in their nuclei, while males always have a single X and a single Y in theirs. In other words, the Y acts like a dominant gene for maleness, but one that can only pass from father to son, and the X acts like a recessive gene for femaleness, which can be inherited from either parent. Stevens and Wilson had made the first ever correlation between an inherited trait and a particular chromosome. Edmund Wilson worked at Columbia University in New York. In the same building was another lab that within a few years became the worldwide mecca for geneticists. It belonged to zoologist Thomas Hunt Morgan and his student colleagues. From about 1910 to 1930, they formed a unique and highly productive research team. Through a series of ingenious experiments, they explored and mapped the chromosomes and discovered within them the secret hiding places of Mendel's hereditary factors. They worked in what is now this teaching lab, which back then was a slightly larger room. In this limited space, Morgan and his students advanced genetics from the level of the chromosome to the level of the gene. They did it with the help of a new experimental partner, the tiny fruit fly, or Drosophila. It is far more efficient than Mendel's pea plants. Its reproductive cycle is only 2 weeks, as opposed to a year. It breeds prolifically, and huge numbers can be grown in a small container. As well, its chromosomes are particularly easy to study under the microscope. But unlike pea plants, fruit flies didn't come in different varieties. So Morgan and his students set out to produce their own, without much success. Then a breakthrough finally occurred. They noticed a male fly with white eyes instead of the usual red. When they crossbred this mutant male with a normal red-eyed female, all of their offspring, the F1 generation, had red eyes. According to Mendel's theory, this meant red was the dominant trait and white the recessive. But when the offspring mated among themselves, the next, or F2 generation, showed a pattern that Mendel would not have recognized. About half the males, but none of the females, had white eyes. According to Mendel, gender should not have mattered, and in this generation, about a quarter of both the males and females should have white eyes. But that is not what Morgan and his students found. However, they were able to explain this exception to Mendel's principles by adding in the newly discovered sex chromosomes. They concluded that the white eyes in half their male fruit flies was indeed a recessive trait as Mendel understood it, but one that is located on an X or female chromosome. To show why this is so, let's examine the famous case of Queen Victoria and her offspring. Many of her male descendants inherited a disease called hemophilia that causes a catastrophic tendency to bleed and usually an early death. It only affects males and is caused by an abnormal gene within the X chromosome. Victoria, being a woman, had 2 X chromosomes in her cells. One of them carried the recessive gene for hemophilia. The other carried its normal counterpart, which functioned properly and saved her from the disease. Her normal X chromosome segregated out during meiosis and was inherited by 6 of her 9 children. The other 3 were not so lucky. They ended up with her other X chromosome, the one with the gene for hemophilia. Her son Leopold, like all males, inherited a Y chromosome from his father. So unlike Victoria, he didn't have a second X chromosome to compensate for the recessive hemophilia gene. He therefore had the disease.
Hex is a hex.
Alice and Beatrice also inherited their mother's recessive gene, but they also received a normal X chromosome from their father, which saved them.
Thanks, Daddy.
So they were carriers like their mother. Their sons too had a 50-50 chance of inheriting the disease gene and having hemophilia, like unlucky Leopold. This is the same pattern of inheritance that Morgan noticed in his white-eyed fruit flies. Both traits show up only in males, and in more cases than would be expected with an ordinary recessive gene. This pattern of inheritance is called X-linked recessive. Through dynastic marriages, Victoria's X-linked hemophilia gene eventually found its way into many of the royal families of Europe.
We are not amused.
Morgan and his students discovered other X-linked traits in their fruit flies. Through crossbreeding, they were able to combine 2 such traits together in the same fly. As they further bred these double mutant flies and studied their offspring, they noticed that pairs of mutations sometimes traveled together down through the generations. This could only happen if the mutations were located on the same chromosome and therefore stayed together during meiosis. Eventually, Morgan and his students found 4 such groups of linked genes corresponding to each of the fruit fly's 4 pairs of chromosomes. They also found that 2 mutations that are linked together over several generations can eventually split apart and segregate out into different offspring, like totally independent genes. Here is what happens. Before meiosis, the 2 mutations are on the same chromosome. Then during meiosis, one crosses over to the matching partner chromosome. The 2 chromosomes then segregate out and end up in different germ cells. These crossovers take place during that early phase in meiosis when the 2 matching chromosomes, one from each parent, come together to do the genetic tango. When 2 opposing threads overlap and touch each other, they both come apart at that point. The 2 loose ends then switch sides and get reattached. What broke up the linkages in the fruit flies was that eventually a crossover point happened to fall between the 2 mutations. Crossovers ensure that each of the chromosomes that emerge from the genetic tango of meiosis and segregate into separate germ cells is a unique combination of the original 2 dance partners, and that each such germ cell is a unique combination of genes. Morgan and his students had no idea what a gene consists of and no way of detecting its presence. But the fact that genes cross over during meiosis gave them an ingenious way to pinpoint their locations within a chromosome. They knew that if 2 mutations are linked, their genes must be in the same vicinity on the same chromosome. They also knew that when they segregate apart, a crossover point must fall between them during meiosis. These points can occur anywhere along the chromosome. So they reasoned that the further apart the 2 genes are, the more likely it is that a crossover point will fall between them. Conversely, the closer the 2 genes are, the less likely this is to occur. Morgan and his students used this relationship between the distance separating 2 genes and the chances of a crossover splitting them apart to figure out where on the chromosome these genes are located. First, through crossbreeding experiments, they discovered a number of genes that were linked together in a series of pairs. They were therefore all on the same chromosome. Then, through further matings, they tested each linkage for its tendency to break, which told them how likely it was for a crossover point to fall between the 2 genes. From these numbers, they were able to work out the relative distance between each pair of genes. Through a simple puzzle-solving exercise, Morgan and his students were then able to line these genes up in their proper sequence and distance from each other. They had created the first gene map of a chromosome. Eventually, Morgan and his students mapped out 4 groups of linked genes, one for each of the fruit fly's 4 different chromosome pairs. But with the exception of the X chromosome, they were unable to identify which group of genes belonged to which chromosome. That changed with the discovery of giant chromosomes within the saliva-producing glands of young fruit flies. When stained, each displays a distinctive pattern of bands, which allowed scientists to match each chromosome with its corresponding gene map. In 1933, Morgan became the first geneticist to win a Nobel Prize.
We are the fruit flies that Morgan kept in jars. He won the Nobel Prize, but we're the real stars. Check out the chromosomes in our saliva glands. They're awfully hunky and they have such gorgeous bands.
Oh, he'd be nowhere without our quick birth rate.
Or the way we strut our stuff when we mutate.
And then the color of our eyes becomes distinct. That's how we showed him that a trait can be X-linked.
We know we're just insects, we can't match their intellects, but we're superior to them at sex. We're fruit flies, not barflies, not gadflies, please realize we're the ones that help them get so wise. Our brains are buzzing, they never take a nap. We gave them full instructions for the first gene map. Just follow what goes on when genes get recombined. Then you will see how chromosomes have been designed.
During meiosis, it's sexual hijinks.
Crossing over, trading parts across the links.
Genes that were once your mom or dad's alone, now joined together in a single chromosome. We know we're just insects, we can't match their intellects, but we're superior to them at sex. We're fruit flies, not barflies, not gadflies, please realize we're the ones that help them get so wise.
Morgan's achievements with fruit fly genes, together with the rediscovery of Mendel's laws, led to a popular obsession with genetics. It was now widely believed that all human traits are determined by genes, not just simple physical traits like eye color, but complex behavioral ones like intelligence. A related belief also took root. Just as crops and animals can be improved through controlled breeding, so too can humans. A philosophy known as eugenics.
And of course there are two ways you can do this. One is you can select people that you think have these better qualities and breed them, or encourage them to breed. Or you can take people who have what you believe are undesirable qualities and stop them breeding.
Eugenicists found the latter much easier to do. In 1910, a eugenics records office was established at the Cold Spring Harbor Lab on Long Island to identify genetically defective families. Over the years, hundreds of thousands of genetic surveys were collected and analyzed. Some diseases did show Mendelian patterns of inheritance, indicating they were caused by a single gene. But the eugenicists were more interested in tracking undesirable social traits like poverty and criminality. Although these so-called degenerate traits sometimes did run in families, it was a fallacy to conclude, as they did, that such behaviors were caused by genes.
You can inherit things from your parents in ways that are not genetic. And the eugenicists tend to play down those environmental effects. They tended to play down the social effects that lead people to have antisocial behaviors.
The eugenics movement coincided with the peak years of immigration to America. Many of these newcomers came from Southern and Eastern Europe and were looked down upon by the established population, who were mainly of Northern European stock. The eugenicists provided a pseudo-scientific pretext for these prejudices, which they shared. In 1924, the head of the Eugenics Records Office persuaded Congress to pass a new Immigration Act that effectively closed the gates to these new immigrants and their supposedly dangerous genes. The eugenicists also promoted the involuntary sterilization of the unfit.
And over a period of maybe 20 or 30 years, some tens of thousands of American citizens were sterilized, mainly, of course, in institutions.
But it was in Nazi Germany that the eugenics movement was forever discredited. In this 1930s propaganda film, the message is that the mentally retarded and other defectives, such as this Jew, described as a cunning agitator, are a reproductive and financial plague on society. The first step towards the Holocaust was the eugenic murder of people such as these. It was the tragic finale to the darkest chapter in the history of genetics.
Cracking the code. Genetic futures will be foretold. Cracking the code. Genetic power about to explode. Cracking the code. Genetic mysteries to unfold. Cracking the code. Genetic secrets will be told. Cracking the code.